you for being here with us today. So I'm really excited to give you a really good presentation on biomedical engineering and how that and genetically engineering DNA into a new species and the benefits of it. So what I have here, first of all, we have 3 visual aids, and we also have a reference guide to see if I need to go on anything or if I go off track or that sort of thing. So first of all, what is genetically engineering, and any thoughts on that? Yeah. Okay. Yeah. Yeah. So genetically engineering is the process of extracting DNA through like a syringe or any kind of like a needle. And DNA can be found in bones. It can be found in fossilized, prehistoric, amber, which is also like mosquitoes or bugs or anything like that. That's been fossilized for thousands and thousands of years. We extract that and then we computer engineer and format that into a new sequencing data. And then, like for instance, lab rats or mice is very common to genetically engineer because they carry a lot of diseases, and to weaken those diseases, we genetically engineer their DNA into a new genetically engineered sequence. So the process is very long, and it can take a long process to format. So, and it is very important because we have health benefits, and it includes many trials and many data sequencing through the labs. So when we do health trials, a lot of people that have cancers, a lot of people that have diseases, autoimmune diseases, lifelong diseases, we take them to trial and we test our newly formed product, and then we see if our new medicine that we have from the genetically engineered DNA can really help benefit them to either cure the disease or slow down the disease or help them any kind of good way. So also I have 3 visual aids that can help us, just to go in more in depth of what I'm trying to really talk about. So what happens when we do it? Like, what will happen when we do it? Society will get curious about what we're trying to do. What also will happen when we do it is when we extract the DNA, it not only goes into like an incubator, but it goes into a light surfaced, like area, kind of like a heat lamp for dogs, but it's for organisms to promote growth and to have a safe environment to promote what we're trying to develop. So we do that in a process of 60 months to a year, or less, depending on how fast we track of what we're trying to develop and things like that. And so also like I was saying, mice has carried a lot of diseases. The Black Death was formed from mice in the past. And so what we do that is we extract their DNA, we take it to the lab, we genetically engineer it to a healthier sequence DNA, and then we essentially we make that mice so it's not so lethal and carries so much diseases, and it can help a lot of things like that. Now the ecosystems, plant life, that is also another healthy benefit of genetically engineering DNA, as we can extract the new, because DNA carries billions and billions of strands, and a lot of times when we genetically engineer DNA, we need to fill in the gap. So we get like a frog DNA, we put it into the new DNA, and then essentially if we do form a new species, whether it's a really small organism or a newly formed genetically engineered rat, we could use that new organism's DNA and extract into medicine to cure for the like the harmful substances like plant matter, a lot of ecosystems, green life plant, like the healthy plant. Like if you're a botanist, you want your plants to be healthy, and so a lot of times with that, we can use that as fertilizer and we can make the plants healthy. So there's a lot of ways we can do this. And so genetically engineer into a new molecule form that is bonded with new elements, to sort of speak, society will prospectively cure unknown genetic diseases. So, and again, these are all comes with like my body paragraph, what I'm trying to say, like the new newly bonded, molecules can help with genetic diseases, especially for genetically engineering DNA into a new medicine. Because a lot of humans carry genetic diseases, whether it's like HIV or anything that affects the genetic DNA, and so that is a really good possible outcome. The process is a long process, like I was saying earlier. Other diseases can be cured in cancers, maybe slow down. Agriculture may improve over time through healthier plant life, like I was saying. Now, the money making in this process can be long, as a lot of people can be skeptical of what we're trying to do. A lot of people that are saying, hey, is this going to really benefit society, or is this going to be harmful to the other like deer, rabbits, elk, because a lot of times when we genetically engineer, new DNA, it becomes harmful because it could release deadly toxins. It can release, it could release carbon monoxide. There's a lot of things that we don't know. And so this is a picture of, what it'll look like if we genetically engineer DNA into a lab. I don't know if you guys can see that online. And then this is another visual aid. If you're interested in being a biomedical engineer, this is what essentially you'll be doing if you're working into a lab. Okay. And then we also have a data statistic chart of what happens when we genetically engineer DNA and we test and we go through different testing. It kind of looks like this. And so essentially what we do is we do a lot of testing on various animals, like I said, rats, mice. Then we see the reactions of what happens when we test that new medicine into the rat and see what kind of toxins it releases, what kind of, chemicals, chemical bonds, anything chemical is a form of reaction. And so when we put it into plants, essentially it's something like this. And so we genetically, speaking, this is the statistic chart of what will look like as well. And another form of visual aid I have for you guys, and let me just check my time, is this. So this is a frog, and we use frogs to fill in the gaps for many DNA sequences. So this frog, it says, welcome friends, he's a very happy frog. And frogs can carry diseases. There's a lot of deadly toxins in frogs. There's a poisonous frogs that are on the surface of the skin, actually, down by the Amazons. So a lot of genetically engineering, DNA can help with the health of humanity and society. And so this happy frog will be happy forever if we promote our DNA sequencing. So now lastly, will it improve our health in both in humans and ecosystems? Like, what do you guys say? Will it improve our health? Will it really be what society wanted in genetically engineering DNA? And if we do form a new species, it could be something very, very good and, interesting. And it can be a healthier rabbit instead of a poisonous rabbit, so to speak. So what do you think? Should we do it? Potential risks can include a mating of a new developed species that is harmful to the environment. So like too many to handle, it can multiply and it could take over, or it can release deadly toxins through what we don't know. Like COVID was an accident. It got all over the world. It could be similar to this, how the toxins can release all over the Amazon that we don't know about. So there's a lot of yes and no answers, but overall I think it's a very beneficial way of society to come by, and I think it's something that we should all prosper and looking for a healthier plant bio life for the future to come. So I want to thank you guys. Thank you guys for being here. Like I said, if you didn't see me on the board, my name is Professor Duncan, and I have a PhD in biomedical engineering. So if you guys have any questions, reach out, and I want to appreciate your time. Thank you very much. Thank you. Yeah, thank you very much.